Welcome to today's Explainer. Today, we are diving into one of the most fascinating and, let's be honest, kind of terrifying phenomena in microscopic biology. We're talking about positive-sense single-stranded RNA viruses. But I don't want you to just think of them as simple pathogens. Instead, I want you to picture them as cunning, microscopic cat burglars pulling off the absolute ultimate cellular heist. Seriously, they break in, they hijack your cellular machinery, print their own loot, and then escape without ever tripping the alarms. It's wild!
So here is our heist blueprint for the day. First, we'll meet our culprits. Then, we'll look at the engine they use to replicate, see exactly how they bypass our security, examine their invisibility cloaks, and finally, witness how they completely hack our genetic code. Let's get into it.
All right, kicking things off with phase 1 of the operation. Meet the RNA invaders, the true master infiltrators of the biological world.
Now, it really begs the ultimate question, doesn't it? How exactly does a microscopic, naked strand of genetic code actually manage to hijack your entire body? We are talking about a simple piece of RNA, with literally no cellular machinery of its own, taking down incredibly complex human biology.
What's super interesting is where these specific culprits sit in the grand virology landscape. If you look at group 4 right there, what makes positive-sense single-stranded RNA viruses so incredibly dangerous is that their genome acts exactly like messenger RNA. The very moment they cross the threshold into your cell, they don't even need to translate their code into a different format. They arrive absolutely ready to be translated into viral proteins immediately. They literally hit the ground running.
And you know, we aren't just talking about one isolated family of viruses here. This is a notorious rogues gallery. We're talking about flaviviruses like the Zika virus, coronaviruses, picornaviruses. Now, they all target vastly different hosts and cause completely different diseases, but they all utilize this incredibly efficient shared strategy of the plus-strand RNA genome.
Which brings us right to phase 2, the viral replication engine, or, as I like to call it, the 3D printer of doom.
Think about it. A burglar can't exactly carry everything they need in their pockets, right? Sometimes they have to build their tools on site. Well, for these viruses, that tool is RDRP, or RNA-dependent RNA polymerase. Simply put, RDRP is a viral enzyme that catalyzes the synthesis of RNA from an RNA template. Because human cells just don't normally copy RNA from an RNA template, the virus literally has to code for its own custom 3D printer to churn out millions of copies of its genetic loot.
Chemically speaking, this relies on a totally brilliant mechanism. If you look closely at those aspartate residues labeled down there on the bottom left, they act perfectly to position 2 magnesium ions, those little red spheres you see. This 2-metal mechanism is brilliant. The magnesium ions deprotonate the 3' OH group. Basically, they strip a proton away to catalyze the reaction, stabilize the transition state, and add a brand new nucleotide to the growing RNA chain. It is molecular engineering at its absolute finest.
But hey, we humans aren't entirely defenseless against this heist. Knowing exactly how this polymerase engine works means we can throw a massive molecular wrench directly into the gears. Look at this structural map of the SARS-CoV-2 RDRP active site. See that green structure right in the middle? That's remdesivir. It's an antiviral drug specifically designed to mimic a natural building block. The virus mistakenly incorporates it into the growing RNA chain, and it physically jams the machinery, stopping that 3D printer dead in its tracks. So the crucial takeaway here is this comparison. Natural viral replication uses those native nucleotides and magnesium ions to rapidly churn out copies. But with an antiviral treatment, like remdesivir, we're actually exploiting the virus's own rapid greed. We give it a faulty puzzle piece that jams the RDRP machinery and halts RNA synthesis entirely. Basically, we stop the heist by breaking their favorite tool.
Moving smoothly into phase 3. Bypassing cellular security, the fake ID.
Okay, to really understand the hack, you first have to understand the security system. Normal, healthy cellular mRNA requires some serious processing. It needs a 5' cap, which is a modified nucleotide at the very front, and a 3' A tail at the back. That 5' cap is absolutely critical. It acts as the cellular password. It proves the RNA actually belongs there, it protects it from getting degraded, and it signals the ribosome to start translating it into proteins.
So, I want you to put yourself in the shoes of a picornavirus or a flavivirus for a second. What if you just don't have that cellular password? You definitely don't have the cellular machinery in the nucleus to add that pristine 5' cap yourself. So how on earth do you convince the cell's ribosomes to read your code without getting instantly shredded by cellular security?
Simple. You use a fake ID, and in virology, that fake ID is called an iris, or an internal ribosome entry site. It's this highly complex, folded RNA structure that enables cap-independent initiation. Instead of needing that cap at the very beginning of the strand, the iris allows the virus to initiate translation right smack in the middle of the mRNA. This completely flips the initiation process on its head. While canonical, normal cap-dependent initiation happens at the top. Just take a look at the purple sections below for picornaviruses and flaviviruses. That complex green folding right there, that's the iris. It perfectly mimics the shape and the chemical signals needed to grab a ribosome and assemble it directly onto the RNA strand, entirely bypassing the need for a 5' cap. It literally just skips the security line.
All right, we are on to phase 4, building an invisibility cloak, the safe house.
You see, many of these viruses, particularly coronaviruses, don't just float around freely in the cellular fluid where they could be easily spotted by the host's immune system. No way. They're much smarter than that. They actively remodel the host cell's internal architecture. They hijack the membranes of the endoplasmic reticulum, the ER, and they actually warp them into double membrane vesicles, or DMVs. These basically act as secure safe houses that completely evade the host immune response, physically shield the viral RNA, and dramatically increase replication efficiency by concentrating all their necessary ingredients into one tiny hidden little bubble.
And finally, the grand finale of the viral heist, Phase 5, hacking the genetic code, the master override.
One of their wildest tricks here is called ribosomal frame shifting. Think of the ribosome reading RNA kind of like a needle playing a vinyl record, reading in sets of 3 tiny grooves called codons. Well, a virus can actually encode a slippery sequence, a really specific stretch of nucleotides that literally causes the ribosome to slip and shift back exactly one space, a -1 frame shift. It's exactly like physically bumping a record player so it skips back a fraction of a beat, which completely alters the reading frame for the rest of the track.
Looking at this chart of mind-bending hacking mechanisms, focus specifically on that ribosomal frame-shifting row near the bottom. By intentionally forcing the ribosome to slip like that, a virus, like a coronavirus or a retrovirus, can literally produce 2 entirely different proteins from the exact same single stretch of mRNA. It absolutely maximizes their genetic payload without taking up a single bit of extra space. It's incredibly clever.
But the absolute masterpiece of RNA hacking? That belongs to the coronaviruses. It's a crazy trick called discontinuous transcription. Instead of just copying the whole genome from start to finish like a normal process, the virus literally jumps around, creating nested, smaller snippets of RNA.
Let's actually trace these molecular gymnastics step by step. First, the rRDP synthesis begins normally down at the 3' end of the genome. Second, the polymerase runs into a very specific stop sign called a transcription regulating sequence, or TRSB. Third, and this is the truly crazy part, the polymerase actually detaches with the newly synthesized strand, floats away, and physically jumps to a matching TRSL sequence all the way at the 5' end of the genome just to finish the job. Step 4, the result is a whole suite of nested subgenomic mRNAs, all sharing the same ends but containing totally different middle instructions.
So you might be wondering, why go through all that trouble to jump around the genome? Well, as you can see quoted here from the source material, this jumping mechanism allows for high rates of recombination. Because the polymerase is constantly detaching and reattaching, if 2 different coronaviruses happen to infect the same cell, the machinery can easily just jump from one virus's genome straight to the others. They mix and match, they swap genetic material. This is what allows them to mutate and evolve at terrifying, breakneck speeds.
And honestly, that's what makes us all so incredibly captivating and just vital to understand. We are in a constant microscopic arms race with these things. We design a brand new lock, like remdesivir, and they eventually evolve a new pick. They really are the ultimate biological cat burglars. So I'll leave you with this to chew on. As these plus-strand RNA viruses constantly evolve new ways to hack our cellular security, recombining and frame-shifting their way past our defenses, what will their next masterpiece heist look like? Thank you so much for joining me on today's Explainer. I really hope this helped map out the brilliant and devious world of viral replication for you. Catch you next time.